The next speaker will be Dr. Jennifer Lynch. Dr. Lynch is a toxicologist and an analytical chemist who's been at NIST for many years. And she's also the co-director of the newly established Hawaii Pacific University Center for Marine Debris Research. Jennifer, do you need this, or are you going to stay?
I'll stay.
Advancing, reverse, and the pointers there.
Great, thank you. What an honor to be here today at the National Academy of Science, Engineering, and Medicine, and to be with all of you talking about this topic that is near and dear to my heart and building a career on it. It's talking about polymer identification. So polymer identification is incredibly important when we're trying to measure plastic pollution in the environment or in samples. And that's because when you look here at the continuum of different plastic particle sizes, when you're dealing with this down here, oftentimes it's hard to tell visually, is that particle plastic or is that sand or hair or some other kind of material? And so polymer chemical measurements to identify the polymer is very important to confirm that what you've got is actually plastics. Another important part of this is to determine when you've got a mixed bag of sample, like a gigantic ghost net that's accumulated many different pieces and parts, we want to know what is the composition of all the different plastics that are inside that sample. And why do we care to know about the different polymers that might be in there? There are thousands of different polymers that are produced. These are only the top 6 most commonly produced, and each one has a totally different chemical structure shown there. And that chemical structure leads to a wide diversity of applications and uses for those. So each polymer is used for something different. We also have totally different waste disposal processes for certain polymers. Some we can easily recycle, and you can see that polyethylene terephthalate, the resin code number 1, and high-density polyethylene are 2 most commonly recycled polymers. And this is based on the chemistry. Stratification of these polymers in the ocean is very different. So if you drop all of these polymers into the ocean, what's going to happen if nothing is holding some down and some up for other reasons, just based on chemical density? The polyethylene and the polypropylenes are going to float to the surface. If something's floating on the surface of the ocean, it can transport much farther distances than a polymer that is known to sink in seawater, which is the polyethylene terephthalate and PVC. They'll stay closer to home if they sink to the bottom. This is a very complex slide. Don't worry, we're going to be stepping through this throughout the talk. But again, it shows the extreme variety of sizes of plastic pollution. We are today focused on the 5 millimeter size class from here and below. And what I want to point out is that the chemical method to identify the polymers of these different size classes is different. We have to choose different methods to be able to chemically identify these different sizes. And even before we can get to identifying the polymers, we need to extract them from the environmental sample and then visualize them somehow. And those techniques are different based on the size. This is a very quick literature search of the different polymer identification tools that people have been using over the last 2 years. And what rises to the top, the most commonly used, is ATR, FTIR, followed by other modes of FTIR, followed by Raman spectroscopy. So those are all spectroscopic techniques for polymer identification. And so those are the most common. ATR, I was asked to put this up, sort of the instrument cost. What does this cost a laboratory to do in money and time? And so this is the money graph where you can see ATR probably wins because it has a relatively cheap instrument cost compared to some of the others. On time, it's extraordinarily fast. You pick the little piece up, you put it on the instrument, within a couple seconds or a minute, you know the identity of the particle. So it's cheap and fast, which is why it rises to the top. However, it does have some limitations. Big picture here, two of the challenges that we have in polymer identification is access to reference library of spectra so that we can take our unknowns and run it against a reference library and know what it is. And OpenSpeccy is here to the rescue. So I'm really excited to announce a lot of different useful tools that this research community can use. This is one of them. The website is there. You can already go online and use this tool. Wynn Cowger out of the University of California, Riverside has developed it. It is a free online open source spectral analysis tool that is specifically developed for the microplastic research community. It contains a growing library of polymers and their FTIR and Raman spectra. And it allows users to upload your data, view your data, clean it with baseline corrections, identify your sample through this library search, and then share that information with the rest of the community. And here's an example. Last night, I uploaded a polyethylene terephthalate spectra that I generated from a consumer good that had the resin code number 1 stamped on the bottom of it. I collected this in Raman. And you can see the top 5 hits there are all polyethylene terephthalate, and they came with very high match scores above 89%. And it actually hit out of at least 3 different libraries, one submitted by Horiba, one submitted by Cabernard et al. 2018, which I believe is out of Sebastian Prümke's lab, and a library that came out of my lab. Another tool that we are working on, and I can't promise this yet, this is simply just a proposal. Hawaii Pacific University, where I sit, has proposed to the American Chemistry Council to create a polymer kit. And the kit would look something like this, like a little tackle box. You open a little bin, and inside you would have 10 to 12 different pre-production pellets of that polymer. And you'd get the kit as well as a USB stick that has FTIR spectra and DSC melt curves specific to those materials, so that labs across the world can all be working with the same polymer materials and can generate their own in-house libraries on these instruments, which is really important to do. If anyone is interested in this kit, they're like, wow, that would be really awesome. We are tallying the demand and the interest. And you can send an email to cmdr@hpu.edu if you're interested. Now I'm going to show you a few examples coming out of our lab at each of these size classes. Why am I talking about the big plastics? Well, because the smaller, inside the blue square, the smaller one does reach down into the microplastics, down to 1 millimeter. So if we look at that, anything that's 1 millimeter or above are things we can pick by hand. We can pull them out of the environmental sample with our hand. We can see them with our naked eye. And we can use just typical routine polymer science methods for identifying the polymers. 2 years ago, we published this study with our colleagues at NIST in Gaithersburg, Maryland, validating the very cheap, easy, fast method of ATR, FTIR to identify polymers of marine debris plastics. And we found that it works very, very well. We actually talked about how well it can do at differentiating the different densities of polyethylene, high density versus low density polyethylene. Since then, we've decided, don't trust us. Don't trust us on that one. So we want to get that message out there. And the reason is, if you start using a different method, differential scanning calorimetry, which is really good at differentiating low-density polyethylene from high-density polyethylene, shown here, see their melting temperature is quite different from 106 up to 135, so you can differentiate them very well. Once we started doing this, we started recognizing that we were a wee bit wrong. And So this is our data to show that we were a wee bit wrong. If you take 16 different pieces of Hawaiian beach debris from different weathering codes, and all of these were identified as low-density polyethylene on ATR FTIR. So we thought we had them. Then you put them through DSC, and what you find is that we were right when they were mildly weathered, but we were 100% wrong when they were severely weathered. So ATR is not reliable for differentiating those when we're talking about weathered polyethylene, and the weathering really affects the ATR FTIR spectra. Recently, we've adopted this four-step workflow. Now, this is tedious. This takes time, but it gives you the most important, the most concrete information about what your plastic particles are made of. And Chevron Phillips actually opened their door and showed us this method. So you can use this workflow to reverse engineer plastic products from the shelf in a grocery store, or you can use this method to forensically reverse engineer plastic pieces that are on the beach. So what you start with is getting used to this. There we go. What we start with is the ATR FTIR, and okay, that's shown here, and you place your piece. You want to get at least two measurements, one on each side, so that you're identifying the layer of polymer that's just on the outside of that particle. That's all ATR can do. It can only go down about 1 micrometer of where you put it on the instrument. The next step is to take that piece and press it into a thin film using a heat press and put it through total transmission FTIR. That way, the FTIR signal is transiting through the entire cross-section of that piece. And if these are the bands on your spectra off the ATR, then all of a sudden you start to see these bands when you're doing the total transmission. You know you have more than one polymer inside that piece of plastic. Then you take a cross-section and you put that on the differential scanning calorimeter. You can tell the difference between the polyethylenes, and you can also understand additives that are in there. It gives you a wealth of information. Then you cross-section the plastic piece, you put it in a microscope vise, and you look under a microscope at the different layers. I'm going to show you an example of what that looks like next. This is a snack food tray that was found as marine debris on a Hawaiian beach. When we put it on the ATR FTIR, we scanned the inside of the piece, and it turned out to be ethylene vinyl acetate. If we flip it over and on the bottom of the tray, we analyze it by ATR, we see polystyrene. If we work our way through this process, what we end up with is the cross-section picture under microscope and all the different layers in there. And then we can see that there were actually 3 different co-extruded layers of polystyrene. There's an EVA tie layer, there's an ethylene vinyl alcohol, a layer of polyethylene, and a layer of EVA blend with polyethylene. So the point here is plastic pollution comes from plastic that is a 3-dimensional product that we use, right? And most products that we use currently today are not just a single polymer. They're multi-layer composites. What we revealed while using this with our Hawaiian marine debris samples, more than half of them were made of multiple polymers, and 73% of those would not have been revealed by ATR FTIR alone. So multiple methods are needed if you want that level of detail. I'm going to move on to another example, moving into squarely the microplastics, where we're looking at abiotic samples of beach sand. We need a physical and chemical way to extract the plastics out of the sand. We visualize with a microscope, and then we use microspectroscopy to analyze those particles. This is the plastic that we're facing on the windward beaches of the main Hawaiian Islands. This is very near my laboratory, so going out and getting samples is quite easy. This is the beach at a National Wildlife Refuge, and all those colorful things are microplastics. We use no innovative tools for sampling. It's a quadrat and a shovel. Our extraction method was engineered and designed by Ray Ivazian, who is an undergraduate student in my lab. It's a wheelbarrow. I did it again. It's a wheelbarrow with a pump underneath and a car battery. And it cycles water through the wheelbarrow and over a tray. And what we do is there's typically a tray sitting in here. We put 2 buckets of sand, 2 buckets of seawater, start the pump flowing, and what floats out of the sand is most of the plastics that are on windward beaches. I'd like to point out that he has the whole scheme for creating one of these on his website at seed.world. This is what we end up with, the 2 compartments from his buoyancy separator device. We have a lot of plant matter that floats to the top, and that is a drag when it comes to science. Get that out of there, right? And then we have this beautifully cleaned sand go back on the beach. That sounds great. But now we have to deal with this mess in the lab, and how do we do that? He's ingenious. He came up with the trash time machine. It's basically a vacuum pot. And I don't know how he came up with it, but it's brilliant. You take this mixture, you put it in a pot, you add seawater, you put a cap on it, you pull a vacuum at 1 atmosphere. That pulls all the bubbles out of the plant matter, waterlogging the plant matter. Plant matter is more dense than seawater. You release the vacuum, the plant matter plummets to the bottom, the plastics stay floating. Amazing. So it's not perfect, but this is what it looks like at the end, and we're much happier in the lab with that. Then we separate with sieves, ending up with 8 different classes of microplastics, and I'm going to show you data that we just recently gathered from this size class, above 63 micrometers. So we sent the 63 micrometer sample off to Agilent, and they placed it on a microscope slide, put it inside their LDIR 8700 instrument, which is a spectroscopy tool that chemically maps what is on the microscope slide. So this is a chemical image map of each dot is a different particle that was in this plastic compartment. And every color is a different polymer or material that the instrument was detecting. And so you can see in the plastic component, the majority is polypropylene, showing up as these light blue specks. We believe that there should have been more polyethylene, which is down here near the 5% range. Anyway, polyethylene there. We expected it to be up here too. So we have some more validation of this method, but what's really cool is the image that you can see. What's also really cool is the spreadsheet that it spits out of each individual particle as a row, its size to answer Bart's concern about the size distribution of the particles, its material identification, and the match. Then we can take all of this information and we can show that within our plastic compartment of our buoyancy separator and trash time machine, that we're getting a lot of plastic in this compartment. In the plant matter, we still get a little plastic, which is shown in blue, but the majority of it is natural debris. And in that clean sand, voila, we did it. We have clean sand. So we're happy to say that. Now I'm moving on to biotic. I just showed the abiotic. Now I'll show you biotic. And in larval fish, what we've been doing is really time-consuming micro dissections of larval fish. The fish are the size of my pinky fingernail. Can you imagine dissecting the gut out of that and then dissecting the plastics out of that little tiny gut? Not easy. We don't want to keep doing that, okay? We do that under a microscope, and then we use both. We used micro FTIR and micro Raman for this work. This is published in the Proceedings of the National Academy of Sciences, and this shows all the different fish taxa that we looked at. And I'll just point your eye to the flying fish larvae. 15% of those were ingesting some kind of microfiber or microparticle that appeared to be plastic. And the spearfish, swordfish, and triggerfish, about 8 to 10% of those were ingesting anthropogenic particles. This shows sort of the workflow. We have the baby, the That's a spearfish. This is a microfiber that was pulled out of its gut. We put it on the ramen, and we also sent it off to Thermo, and they put it on their microscope FTIR for us. And what's really nice is having the complementary data that both of these, for this particular piece, pointed to polyethylene terephthalate, which is polyester fabrics. So we felt very certain that this one was polyester. We don't want to do those micro dissections anymore. We want to take the little larval fish, plunk it in a test tube, digest the fish away from the microplastics. I wish. It's going to be really hard. Before we dive into the samples, we wanted to take the polymers that we routinely see in Hawaiian debris and put them through these chemical techniques. And so this is the master's project of Jenna Carr. And by the way, everything I'm showing today is mostly unpublished. We're still working on it. So this is hot off the press stuff. Jenna took all these polymers in triplicate, put them in test tubes, put them through incubations of these different chemical solutions for storing the fish and digesting the fish, measured the mass fragmentation, size, and FTIR of each of the polymers after doing that. And these are her results so far. We still have a few trials to do, but basically everything in red, you don't want to use that chemical for that polymer. Everything in orange, you probably don't want to. And nitric acid was one of the most common digestion methods for trying to do this. And it works okay for those heavily produced polymers, but when you get down into the other polymers, you really don't want to be using it. So this gives us a chance to show side-by-side compatibility of those chemicals with the polymers we're interested in, so we don't underestimate the polymer concentration in the fish. Moving quickly, because I'm running out of time, into nanoplastic realm, I want to start by saying we don't have any data. Okay, I don't want to get anyone's hopes up. This is going to be challenging, and it's unknown yet how we're going to extract these particles out of samples. We need things like scanning electron microscope to see these particles, and how do we identify the polymers? We're all not sure, but we have some promising techniques that have been suggested. Notice this gigantic word, proposal. This is a proposal from within my federal agency, National Institute of Standards and Technology. We are perfectly poised to help with this, this field of study, and we have quite a few researchers within NIST that are very interested to help. We're just all overstretched and burdened with everything else we have going on. So we put in a proposal for internal NIST money to help support us so that we can move this field forward. And these are our ideas. We're welcoming you to tell us more ideas, to ask us, this is the kind of thing we need, please help us. So we're looking for feedback. We just turned in this proposal that talks about producing nanoplastic standard materials, so of different polymers. We could potentially produce those in our nanofabrication facility. So NIST is known to produce nano-sized materials. So we're talking about maybe using that facility to do it, or take larger pieces of plastic and expose them to harsh weather conditions in the NIST weathering sphere and watch nanoparticles fall off of them. So those are two possible ways. We definitely want to help with detecting, characterizing, and quantifying nanoplastics. Charlie Camp has helped develop this very fast scanning Raman microscope that could be very useful for this field. And I'm interested in pyrolysis GCMS, which are promising methods. Then I hope to help test methods for the separating, purifying, and concentrating. Nothing innovative here. These are filtering, sieving, sonicating kind of methods, but they've not been tested for this application. And finally, performing in vitro toxicology studies. We have scientists at NIST that are interested in developing 3D models to look at the translocation of the particles across tissue barriers. I've just presented about 8 different projects, all of which have many, many co-authors. So huge thanks to them. Huge thanks to my family that has sacrificed a lot. My daughter is in a hotel room down the way, traveling all the way from Hawaii, and that's her. And if you're interested in following our work, we do a lot of posts on social media. So like us, follow us.